Hello again, everybody. Today, we are going to be doing diseases of the cardiovascular and the lymphatic system. In tune with everything else the way I try to do it, we'll talk a little bit about the anatomy of the systems first. And the cardiovascular system, if we talk about the cardiovascular system, composition of the blood vessels and the heart, which is really what they refer to as the pipeline of the body. It's a closed circuit known as the circulatory system. The heart pumps the blood and moves in this closed circuit called the circulatory system. Of course, the function, of the cardiovascular system is to provide the tissue with oxygen and nutrients, and also to carry away carbon dioxide and the waste products. Over here, I took this directly from your book. It shows the circulatory system and a very closely related lymphatic system, and then shows you that most of the time, the lymphatic system, which we're going to talk about, it's more like a return passage of material. And we'll talk about that in a second. Now, you have the heart, and you have done a lot more in your anatomy and physiology course about the heart. But just to give you a brief description, it's a fist-sized muscular organ that pumps the blood. Atria are the upper chambers, the ventricle, the lower chambers. And then you have the pericardium, that fibrous covering, the walls of the heart, epicardium, the myocardium, and the endocardium. And then, of course, we have the blood vessels. Actually, three groups of them: the arteries that carry the oxygenated blood, and the veins. And of course, they carry the deoxygenated blood back to the heart to be reoxygenated. And then you have the capillaries, the smallest vessels that connect the arterioles to the venules. And they can only do that because they have just one layer of endothelium to allow for the exchange of gases. Here is a picture of the heart that's been labeled for you. You can take your time and look at that and see if you can absorb what's there. The lymphatic system is closely related to the circulatory system, like I said, but it is separate. It's the main source of your immune cells and fluids. And normally what happens, the blood will take fluids and cells to the tissue. It is the lymphatic system that will collect that fluid that has been left, filter it of impurities and infectious agents, and return it back into the bloodstream. And that's what this picture was showing you. The blood is taking cells and fluids to the tissue, and it is the lymphatic system that will filter it, purify it, and bring it back to the circulatory system. The lymphatic system is made up of lymph vessels running roughly parallel to the blood vessels, lymph nodes. They cluster at body sites such as the groin, the neck, the armpit, and the intestine, and of course the spleen. All right, that's the anatomy. Now let's see how your system is trying to defend itself, or these systems defending themselves from intruders, organisms trying to invade and cause damage. The cardiovascular system is highly protected from microbial infections. But if organisms enter that system, of course, they have access to every part of the body, because anything that's in the blood is going to be distributed to every part of the body. And for that reason, if something is in the blood, affecting the blood, then every system, every organ system has the potential to be affected. So the bloodstream infections there are considered systemic infections. There are multiple defenses you put up. In the bloodstream, you have about 5 to 10,000 leukocytes per milliliter of blood. Lymphocytes, they're the ones that will kind of produce that specific immunity we talk about. They're the ones that are going to help the B cells to turn into plasma cells, and then those will secrete your antibodies. And of course, phagocytes, they are white cells, and they are very critical to specific and non-specific response. Medical conditions involving the blood usually have the suffix emia. And just to give you a couple of examples, viremia, presence of viruses in the blood; fungemia, presence of fungi in the blood; bacteremia, presence of bacteria in the blood; pyemia, and so on and so forth, emia being the suffix. Now, septicemia is bacteria growing and flourishing in the blood. Now, we just said bacteremia is the presence of bacteria in the blood. It is very important for you to recognize that there is a difference. You may have bacteremia in the blood as a transient situation. You might be brushing your teeth one day, and maybe you're using too harsh of a toothbrush, or you're brushing too harshly. You break the gum. Organisms from in between the teeth can enter the bloodstream. And that is bacteremia, bacteria in the blood. Does that mean you're sick? Not necessarily. It's usually a transient situation, and bacteria will be removed from the blood. Septicemia, on the other hand, you do have bacteria that are now growing and causing damage. This is a disease state. So you are having the bacteria growing there, multiplying, and causing a disease situation. So that is septicemia. Now, the cardiovascular and the lymphatic system are closed systems. And so they don't have normal access to the external environment. In a healthy, normal individual, usually no organisms colonize either the lymphatic or the cardiovascular system. And because there is no opening to the outside, there is no need for guards at the gates. There is no gate to the outside. And so what I'm trying to tell you, normally, no normal flora. Okay, so that is the anatomy and your defenses. Now, we don't have any normal flora there, And normally, we're going to say the next thing we should do is to talk about how we acquire these diseases. Well, what I'm going to do instead is talk a little bit about the diseases, because you can acquire these diseases in so many different ways. An insect might bite and inject the organism into the bloodstream. Or you might have some other infection of another organ system and the organism just gets into the bloodstream. You may have an injury that causes organism to get into the bloodstream and take hold. So there are many different ways for you to get organisms into the bloodstream. And like I said, I will give you a couple of diseases, and then we'll talk about how these organisms are getting into the bloodstream. When it comes to symptoms of these diseases, well, obviously, if organisms are in the bloodstream, you are going to have interference with the proper functioning of the blood. One of the first things that will probably happen is a high fever. And we'll talk about all these different fevers that we generally will have. But the worst part, it is systemic, and you can sort of contaminate all the other organ systems. So it can lead to general destruction of cells and tissue. Okay, so we'll talk a little bit about some of the diseases. Like I did with the other systems, I will talk about a couple of them. I'll try to mention most of them, and then I'll give you a few to work on your own. And so we'll get it basically as much of it out as possible. Now, the one that I chose to talk about is malaria. And malaria has been one of the greatest inflictions, or afflictions, I should say, throughout human history. It is the prominent protozoan disease. We talked a little bit about protozoa in the lab. And it is caused by different species of an organism we call the Plasmodium. It threatens about 40% of the world's population every single year. There is a little incubation period, but then the symptoms set in. And the original symptoms are like malaise, fatigue, aches, pains, nausea, chills, fever, and sweating follow later on. But then almost synchronously, you're going to have your blood cells being ruptured, and you're going to have those chills and fever and sweating we call the paroxysm, closely correlated with the rupturing of the blood cells. A little something of what's going on in there. The malaria parasite, maybe I should talk a little bit about that first, usually enter the bloodstream from the liver cells, where it got it from the mosquito. The mosquito is injecting, it gets into the liver, and then to the bloodstream. But let's talk a little bit about what goes on in the bloodstream. The organism enters the red cell, and I'll show it to you later on, like a ring form. And the ring will eventually develop, and you end up with a lot of merozoites in that red cell. And the merozoites will cause the red cells to rupture, not going to hold it anymore. And then each one of those merozoites can now infect another red cell, a non-infected red cell. What I'm trying to tell you is that it takes a little while for the ring form that just enters the red cell to develop into the merozoites that will cause the red cells to burst open. And that period of time, it becomes like fixed. In some cases, it may be 4 to 6 hours, which means, I'm sorry, I don't think it's that short. Maybe it could be 18, let's say 14 to 18, maybe even 20 hours. In other cases, it could be very, very short period. What I'm trying to say is that the maturation time then will vary depend on the species. And whilst the ring form is developing into that merozoite that will rupture the red cell, once that phase is going on, the patient is seemingly normal. They don't have too much of the aches and pains and all the symptoms. But once the red cells break open, then those symptoms will appear. The fever and all the, what they call the paroxysm, this feeling of nauseousness and probably vomiting and whatnot. And that will happen for a period of time until the merozoites now enter red cells again and start that developmental cycle, and the patient is okay again. Okay? That's what we're talking about, the regularity of the symptoms. And it will depend on which species of the malaria parasite we're talking about. It is that development of what's going on. I can call it the maturation stage, where the ring form is maturing into the merozoites that will cause the red cell to break open. Okay? Within that period of time, patient is seemingly normal. And then red cells break open. It's almost like all hell breaks loose. And so the patient is in a lot of distress for a couple of hours. Now, complications. Of course, the organism is in the bloodstream, in the red blood cell. Remember, we talked about the apicomplexa group of organisms that are intracellular. This is one of them. So it's going to cause hemolysis of the red blood cells, leading to hemolytic anemia. A lot of the debris after the red blood cells break open, cellular debris will accumulate in the spleen, the liver, the kidney, cause them to get enlarged. So that's one of the other complications. If they happen to be in the brain, it's more complicated because the small vessels in the brain become obstructed. Okay, the RBCs, they start to adhere to the vessel wall and the vessels are obstructed. Decreased oxygen in the brain can lead to coma and eventually death. So I said there are 4 species of Plasmodium that will cause malaria. Plasmodium malariae, Plasmodium vivax, Plasmodium falciparum, and ovale. Your book lists a 5th one there, probably an alternate name for one of them. Humans are the primary vertebrate host. And you have, when I was mentioning sporogamy, and schizogamy. Sexual, sexual and asexual reproduction in protozoa. We talked a little bit about the asexual phase in humans and the sexual phase in the mosquito. I'm not quite sure. If you remember, you can always refer, but there is a little picture. It might be a little difficult to read. But like I said, look what happens here. The mosquito is injecting something into the human liver. And from the liver, it gets into the bloodstream. And now it's throughout the system. What happens is that the red blood cell is infected with something you call a little ring form. And the ring form develops and into a trophozoite, mature. And then eventually, a lot of merozoites come out of that one red cell. This is what I call the maturation period. When that happens, that's when you're going to get the symptoms. So each one of these merozoites will now enter another red cell and cause. From the time the ring form enters to the time the cell ruptures, you can have hours where there are no symptoms at all. Seemingly the patient is normal. And so it's almost like a respite for the patient. But once the red cell break open, that's when you're going to have all the symptoms of malaria. And this will go on in the patient's blood for a long period of time. And as the cycle goes on, some of these merozoites can differentiate into gametocytes. So now when the mosquito goes to drink the blood of this individual, it's ingesting maybe regular blood cells that are infected with the malaria parasite, and also gametocytes. It turns out that the gametocytes will not be digested by the mosquito. But instead, in the mosquito, there is another cycle of development going on. And eventually what will happen, the mosquito, because of the gametocytes in there, combining to form a zygote, and then the multiplication and development should take place in the mosquito. That is what they call the sexual phase in the mosquito. And so what comes out of the mosquito are sporozoites that are injected into a new victim's liver to start that cycle all over again. Okay? So basically, that's how malaria, when we talk about the asexual and the sexual phase, you need that mosquito to carry on the life cycle of that organism, so that organism can now be injected into a second individual that can develop malaria. Now, what's going on? The pathogenesis. I told you the invasion of the merozoites into the red blood cells. That's forming the ring. And these will lead to fever-inducing chemicals into the bloodstream. And that's where you end up with the chills and the fever that occur in that cyclic pattern. Red cells will be damaged, leading to anemia, and the accumulation of what they call the malarial product. Remember, when your red cells rupture, there's a lot of debris now in the bloodstream, and that debris can accumulate. And I said the liver, the spleen, sometimes the kidney, and causing an enlargement of these areas. Malaria is transmitted by the female Anopheles mosquito. Now it's controlled in the temperate areas of the world, but it's now kind of restricted in the belt around the equator. Because for a while there, as long as female Anopheles mosquitoes were around, there was the possibility of getting malaria. But now they seem to have restricted themselves in that endemic area around the equator. 3 to 5 million new cases are still reported each year. 90% of these cases are in Africa. And most of the time, the victims are children and young adults. 2 million die annually. In the United States, you have some new cases, but the new cases are generally in individuals that are migrating into the country, or the United States citizens are visiting endemic areas. And so it's not like it's prevalent or it's endemic in the United States, but you do have new cases coming into the country with the immigrants coming in, or people going out to visit these endemic areas. And so they can contract malaria and bring it back. Diagnosis of malaria? Well, it is in the bloodstream. And most of the time, we take a sample of the blood, make a smear, a regular smear, and actually look under the microscope and look into the red cells to see the ring form or the developing trophozoites, or sometimes you can even see the gametocytes in the blood. And most of the time, that's the way we diagnose malaria. There are some newer techniques out in the market. A lot of people, they're still very new, serological procedures out there, not well used right now. Easy to do a smear, a right stain smear, look into the red cells and see the organisms without a problem. And so the newer techniques that are coming out will probably be an addition to using blood smears to diagnose the disease. Because with the blood smears, not only are you seeing the organisms, there are ways to differentiate different species of malaria in the blood. How do we prevent malaria? Well, if you can prevent getting bitten by the mosquito, I think that's one of the best ways to do it. But if you live in those endemic areas, after a rainy season, it's very difficult to avoid mosquitoes. They're going to be there. So they're working on probably doing things to the mosquitoes to make sure that they are not able to carry the organism, or the organism is not able to go through its life cycle in the mosquito. And if they can do that, then that would be great. Now, humans can reduce their risk. They use netting, screens, mosquito repellents. And your book shows you some picture where during the nights, people are actually, and even sometimes during the daytime, depending on what you're doing, you're actually enclosed in netting to prevent mosquitoes from getting to you. Remaining indoors at nights, but mosquitoes get into the house, so you got to be under your nets. And there are some antimicrobial drugs you can use. Quinine, for example, can be used. We talk a little bit about that when we're doing antiprotozoan drugs. Now, the best protection is a vaccine. If you can find a malaria vaccine, that will be the best protection. Now, for decades, researchers were trying to get a vaccine. I think it's 2018, there was finally a breakthrough, and they did come out with a malaria vaccine. Okay, and it is said that the vaccine is very effective, especially in children and young adults, the ones who are hit most or the hardest by this organism. A lot of people going into the area now, I think maybe they're required. I think they're required to have prophylaxis, and the anti-malaria vaccine will probably one of them that will be, they will insist on people having one of those. But anyway, a lot of research is going on to figure out how they can work on the mosquito, not the organism itself, but the mosquito. See what they can do with the mosquito to render them ineffective when it comes to this life cycle, the sexual phase of this organism. If you can prevent that phase, chances are you can have a handle on malaria. Treatment, quinine, chloroquine. That is one of the treatment that has been used for a long, long time. Quinine, which is something they extract from one of the trees out in their area, in the endemic area, has been used long before even people realized there was chloroquine. And so the quinine, or pharmaceutical companies will add other groups to it, like the chlorine group or mefloquine. Those are actually similar drugs with different attachments to them. There is a drug that they have these days, artemisinin. I hope I'm pronouncing it right. Artemisinin. It's a plant compound, and it has proven to be very effective. Okay, and there are some organisms that are resistant to it, but it's been very effective. So what the World Health Organization is recommending that you administer this plant compound together with some of the other antimicrobial drugs, and this will prevent development of resistant strains. The other disease of the blood and lymphatic system I would like to focus on is the AIDS. Maybe some books will cover it. Your book covers it at this level. Some book will say it's a sexually transmitted disease and will cover it there. But it is one of those diseases that's affecting a lot of your immune cells. And because of that, we're going to cover it under the topic of cardiovascular disease. Now, AIDS, most of the people will recognize the infection because of the opportunistic infections that they keep on having, not realizing it was AIDS or it is AIDS. But people come down with sometimes the AIDS defining illnesses like Kaposi sarcoma, Cryptococcus meningitis, MAI infection, PCP, pneumocystis carinii pneumonia, herpes, and those kinds of things. And those things would lead them to start looking for um, the AIDS virus. Now, the symptoms of AIDS are generally linked to two things: the level of the virus in the blood and the levels of the T cells in the blood. Initial symptoms, like I said, you have other things that are going on: neurological changes, the fatigue, the diarrhea, the weight loss, and then the opportunistic infections and sometimes cancer. Patients will end up with pronounced wasting of body mass. They lose weight so fast and so easily. And that's because of the diarrhea, and they don't absorb their nutrients properly. Because the virus is really hard on the GI tract. And some of the most virulent complications are neurological. So, what about this disease? What's causing it? Why is it there? How did it get there? Well, HIV is a retrovirus, and retroviruses have the potential to cause cancer, hence some of the neoplasms that you see in our AIDS patients. And they actually can, they have the capability of altering the host DNA. They can actually redirect the host DNA to do what they want them to do. And you know, viruses can do that. They contain something called reverse transcriptase. That is very unusual for viruses, but that's what they have. They usually have, it's an unusual enzyme, and that enzyme catalyzes the replication of double-stranded DNA from single-stranded RNA. It is a single-stranded RNA virus. And yet for all, because of the reverse transcriptase, it could make double-stranded DNA from that. And double-stranded DNA can now be easily attached to the host DNA and redirect what the host DNA should be doing. Now, that integration into the host DNA, that now is passed on every time the cell replicates. Those new viral genes are passed on to the new cells. It's an envelope virus with a lipid outermost envelope. And then you have glycoprotein spikes. Let me see if I have a picture. I think I have a picture of that. So here you have an envelope, and then you have these glycoprotein spikes. And this is a very nice picture, but because it shows you how the virus now can attach, because this because of the spikes, they can now attach themselves to the host cell. But there need to be some receptors on the host cells on which they can attack. Most of the time, it's the CD4 marker and a co-receptor marker. And it turns out, unfortunately, that many of our lymphocytes, the white cells, not just lymphocytes, but white cells, and some of the tissue cells have these receptors. And so the organism, this virus, can easily attach and enter those cells to cause the damage. Okay, and that's how they enter your system, because your cells have those markers. And it is unfortunate because the HIV will infect only those cells that are supposed to be the cells protecting you from invasion of invaders, parasites coming into the system. And because they're infecting those cells that are your weapons, they're actually destroying your weapons before you even get a chance to use them. Now, this is talking a little bit about the pathogenesis of the virus. And we notice here that the virus enters the mucous membranes of the skin, mucous membranes anywhere, or the skin, and travels to the dendritic cells beneath the epithelium. They grow there, and they don't kill the cells. The cells can shed the virus without being killed. Now, they're amplified in the macrophages, the lymph nodes, bone marrow, blood, and they will actually infect and destroy your T helper cells and your CD4 lymphocytes. Because remember, those are the cells with the markers. T helper cells are the ones, first of all, the T helpers are the majority of your T cells, number one. And those are the ones that will trigger your B cells to differentiate into the plasma cells to give you your antibodies, to secrete the antibodies. So it's like the organism is destroying both halves of the immune system, the one that will give you specific immunity and the other half that will give you the non-specific immunity. And that's what makes this virus so devastating. Transmission, sexual intercourse, the semen and the vaginal secretions harbor free viruses that will infect the white blood cells. And that is, one of the major ways individuals contract the disease. Babies can get the disease from the milk that the mother is feeding. And you can also get the disease. Remember, it can enter the skin and the mucous membrane. So a lot of times, not a lot, but there are occasions when accidents do occur and healthcare workers can infect themselves by injecting the virus through dirty needles. And like I said, accidentally that has happened and individuals have died because of that. But in the outside world, away from the hospital setting, you have a lot of individuals who are drug abusers and they inject themselves constantly. So it's easy for them to transmit the disease if they're using dirty needles to inject themselves. Okay, so that's one way you can transmit. It's since the beginning in the early 1980s, more than 30 million people died. Okay, so about 32 to 38 million people who are infected. There are about 2.1 million new infections that occurred in 2009. And like I said, the statistics are very delayed. Not quite sure what it is right this moment. But due to global initiatives, a lot of it sponsored by the World Health Organization, more people are in the developing world. They are receiving life-saving treatments. It's not, those drugs are not readily accessible in the undeveloped world. And that's where the World Health Organization is coming in to see how they can help to get the drugs to those individuals. How do we diagnose? Well, first of all, you're going to test for the virus. It's not the same as testing for the AIDS, the disease. The viral testing, you can actually test for antibodies specific to the virus in the serum or other fluids, ELISA test, latex agglutination test. Sometimes at home testing kits are also available. These are like screening tests. They're largely accurate tests, but there is a margin of 1% that could be false positive. Sometimes, instead of checking for the antibodies, you're checking for the antigens, meaning the virus itself. Because a lot of times when you stop producing the antibodies, you need to go to looking for the virus itself. In the US, the way they diagnose is you're testing for the virus first, if it's positive, and then you have to meet two of the following criteria. CD4 cells below a certain number. Okay, and the experience of one or more of those AIDS-defining illnesses I mentioned earlier. How do we prevent AIDS? Well, avoidance of sexual contact with infected individuals. A lot of times it's not easy to tell who is infected and who is not. So you're not going to be totally abstaining from sex. It's not a natural occurrence. But you can use some preventative measures. Sexually active individuals should consider that every partner is infected unless proven otherwise. It's almost like saying you need universal precautions in the lab. Protect yourself. Pretend as if every patient has the deadliest of organisms. And so sexual partners should consider, if they don't know, that every sexual partner is a potentially infectious individual. And so protect yourself. Most of the time, barrier protection is used. Okay, especially when you don't know if a person is positive or negative, you need some protection. Avoid intravenous drugs. If you can't do that, at least avoid sharing of needles. And then, of course, some prophylactic drugs are in the market these days. You can use those, okay, and see what's going on there. I think, if I'm not wrong, I heard about something called Truvia that they're advertising these days. If you use Truvia prophylactically, it's more like you are treating before you even contract the disease. There is no cure for HIV, but there are some things you could do. And a lot of people are living these days. They have a very positive outlook in life. Their quality of life is improved because there are some things they can do about it. Drugs that are available, preventive measures that are available. I mean, I have some drugs there. See what happened? We said that the organism produces the enzyme reverse transcriptase. And if we can stop the reverse transcriptase from being produced, then you can probably have a handle on the virus. So reverse transcriptase inhibitors like AZT have been used. There are also protease inhibitors like ATV that has been used. And like I said, although there is no real cure, some people can live not healthy lives. I wouldn't call it healthy lives, but they could have good quality life with AIDS, with the HIV virus. Now, some of the challenges about the treatment, we need to make the drugs available to a greater part of the world, not just the developed part that are privy to the drugs. Make it available to just about everybody. And it's a challenge because to make these drugs and being able to deliver them in an affordable and practical way can be a huge challenge. All right. So now I'm going to talk a little bit about hemorrhagic fever diseases. It's not really one disease, but there is a lot of agents, infectious agents, that can infect the blood and lymphatics, and they end up causing fevers. That's what you call them hemorrhagic fever diseases. Very high fever, some of which are accompanied by internal hemorrhaging. And so the term hemorrhagic fever diseases. Normally, what will happen, viruses, organisms are in the bloodstream and they cause the capillary fragility. Sometimes they disrupt the clotting system, and that would lead to various degrees of pathology and death. All the virus causing hemorrhagic fevers are RNA virus, and they are also enveloped viruses. Okay? So let's see if we can talk a little bit about these diseases. Ebola. I don't know if you heard about Ebola that I think it was, well, it says 1914. I thought it was 1918. But yeah, it's a little longer than that. In 1914, there was an outbreak of Ebola. And actually, the world was not prepared for that. And it was one of the first indications that healthcare is not prepared for a pandemic. And look what happens to, to COVID. Because the Ebola virus, the efforts to control it were lacking, way behind the outbreak itself. It was endemic in Africa, and patients bleed from every orifice. And that's what the death was due to. Bats were thought to be the natural reservoir for the Ebola virus. There is something called Lassa fever. It may be asymptomatic in 80% of the cases, but in others, there are severe symptoms that will lead to death also. And again, the rat is the reservoir for that virus. So it seems like the bats and the rats and those animals are very dangerous to have around, but they are part of the ecology of this world. So we need them there too. Yellow fever, you might have heard of yellow fever, transmitted by the Aedes, not the Anopheles, but the Aedes mosquito. And usually you can have yellow fever just about around the equator belt also. But it's more frequently found during the rainy seasons. When you have those tropical rainforests, you have lots and lots of mosquitoes around. Dengue fever is another one, endemic in Southeast Asia and India. In fact, I was talking to a colleague of mine from India, and he was saying that in India, more people die of dengue fever than people died of the COVID. Well, they have a serious COVID problem right now. Okay. And again, Aedes mosquito. So our mosquito is not our friend at all. There is also fevers that are produced by other organisms and caused by a group of agents that result in the same kind of syndrome, high fever, with or without the capillary fragility. So they do not lead to the hemorrhaging. And so we call them non-hemorrhagic fever diseases. Okay? They're not just caused by viruses. In fact, most of the ones that I chose will be bacterial diseases. And we call it the non-hemorrhagic fever diseases. Brucellosis, if ever you heard of andulant fever or Malta fever, can be caused by mostly Brucella melitensis, but sometimes Brucella abortus and Brucella suis. They actually can live in the phagocytes. Remember what I just said. Sometimes the organisms enter those cells that you need, that those cells that should protect you from the organism. They enter those cells and thrive in those cells. And here's the Brucella organism living in the phagocytic cells. And since they are in the phagocytes, now they can be carried through the bloodstream. Okay, and you can end up with focal lesions in the liver, the spleen, the bone marrow, the kidney, wherever. One of the symptoms is a fluctuating fever, on and off fever, very high, but on and off. And that's how they call the disease undulating fever or undulant fever. Q fever, I might have mentioned this organism when we did rickettsial diseases. And I also might have mentioned an organism called Coxiella burnetii, which is a little different from the other rickettsial organisms we talked about and how they behave. And I said at that point that Q fever organism, Coxiella burnetii, will probably not be considered rickettsial for a long time. And sure enough, it was once considered rickettsia, but not anymore. And with Q fever, and they call it Q fever because it's they're having a rough time trying to figure out what really causes this fever. Q really stands for query, having rough time to decide what's the real cause of this fever. They know it's Coxiella burnetii, but where is it coming from? Many of the other rickettsial diseases, there is some insect vector. Can't figure it out with Q fever because there is no such thing. All they know is that it's a very small, pleomorphic gram-negative bacterium, and the patient ends up with fever and chills and headaches and muscle aches, and occasionally a rash. Many times, most of the people that are at risk, the highest risk, are farm workers, meat cutters, vets, consumers of raw milk, because this organism seemed to invade farm animals. And then it seems like from the meat handlers and whatnot, individual humans can get it too. But they're still not quite sure how humans are infected with this Coxiella burnetii. I mentioned Rocky Mountain spotted fever when we were doing those rickettsial diseases, and it is caused by Rickettsia rickettsii. It is called Rocky Mountain spotted fever because it was first discovered in the western part of the United States. But now, most of you can find them on the eastern seaboard, southeast and eastern seaboard regions. Rickettsia rickettsii is transmitted by the tick, and 2 to 3 days after the incubation, you're going to have sustained fever, again, chills, headache, muscular pain. What's important is that you have this very distinctive spotted rash, and that's why they call it spotted fever. Babesiosis. There is an organism called Babesia microti. This is a protozoan that lives inside of the red cells. It is also carried by ticks, transmitted by ticks. And one of the things that sometimes there is a misdiagnosis with this organism because it looks very much like the ring forms of Plasmodium, the malaria organism. So a lot of times, if you're not careful, you can misdiagnose this disease, especially in the earliest stages of malaria. You'll find a lot of ring forms. And that's what this organism would look like inside of the red cells. One of the things that will help you to differentiate and look for something carefully is the location. You see, with malaria, the endemic area, mostly Africa and some of the areas around the equator. But with Babesia, it's more like the Great Lake region, northeast of the United States. And so if you're looking carefully at where the patient has been, where the patient visited, then you probably will know when to look for malaria and when to look for Babesia. Okay? Now, I think that is what I want to talk about, the cardiovascular lymphatic diseases. And I have a slide here that I would like you to follow the same thing that I did, the same pattern, and try to write a little bit about those 3 diseases. And if you have any questions at all, please let me know so we can discuss them and cover them in whenever time we meet, wherever you want to meet, in the lab or in our office hours, which you just normally you don't even visit. But in any case, I'm going to stop the recording now and we'll continue with our lecture the next time.